Hello, Namaste students. Welcome to One Page Biology. How are you all? I hope you all are doing fine. In today's video, I will be covering one important topic which is called as interface. This particular topic is part of the chapter cell cycle. Topic interface is important for NEET exam as well as for those students who are appearing for 11th standard CBSE board exams or even for other state board exams. So let us understand this topic which is known as interface. Dear students, whenever the cells are going to divide, whether it is a prokaryotic cell or a eukaryotic cell, whenever the cell divides, essentially what happens is the nucleus. Now here I am specifically talking about a eukaryotic cell because the prokaryotic cells do not have a well-defined nucleus. What they have is something called as a nucleoid. So let's just consider over here that this is a eukaryotic cell. In case of a eukaryotic cell, since they have a well-developed nucleus, if you talk about how these cells divide, then there are two things which are going to happen. First, the nucleus is going to divide. The nucleus divides, which is going to be followed by the cytoplasm. So we can say that the cytoplasm is going to divide later. So the first is the nucleus which divides, and the second is basically the cytoplasm which divides. Whenever a nucleus divides, such a type of division is called as karyokinesis. It is called as karyokinesis, and which is followed by, as we said, that once the nucleus divides, then the cytoplasm of the cell is going to divide. So this is known as cytoplasmic division, is called as cytokinesis. Now the question is, before actually the nucleus is going to divide or the cytoplasm is going to divide, the cell has to actually increase in size, or we can also say that the cell has to increase the contents which are present inside the cell, right? Just imagine, for example guys, there is a particular cell. If it directly divides, then whatever the content inside the nucleus is, whether it is the DNA of the cell or whether it is different organelles of the cell, they will split up into half. So if the cell does not double its content, if all the material inside the cell is not actually doubled, then possibly there won't be an equal distribution of the content of the cell. So hence it is very important for a cell to double the content, whatever is present in the cell, whether it is DNA or whether it is different types of enzymes or whether it is organelles. All these content has to actually double in amount so that it can get equally distributed when the cell actually divides into two. So that means such a cell has to prepare itself for division. That means, dear students, a cell has to go through a particular phase during which it is going to duplicate all the material which is present inside the cell. So this particular phase during which the cell actually prepares its content or it doubles its content, such a phase is called as interphase. Dear students, please understand this carefully. As we said, that the phase in which the cell is going to double the content which is present inside the cell, that means in a way that cell is going to prepare itself for cell division. That's why, dear students, interphase is also known as preparatory phase. Always remember. So suppose this is a cell, dear students. Now in this particular cell, almost 3/4 part of the cell, that means almost the 3/4 part of the cell is going to be taken by interphase. So what am I trying to actually tell you is, suppose this is the entire cell is going to divide, and from this particular point, suppose this is the point from where it actually begins the process of division. Let's assume that. So this is the particular point from where the cell enters into the interphase, right? Now, as we said that from this point till this point, the cell is going to actually complete the interphase. So this entire duration of time is going to be taken by the cell to prepare its content, to double its DNA, to form different types of organelle or different types of proteins, etc. That's why, dear students, they say that the interphase is nothing but the most active phase of the cell. The interphase is called as the metabolically active phase of the cell. Once the interphase is complete, then from this particular point, the cell is going to start dividing. But as we said that whenever the cell starts dividing, what is going to divide first? It is the nucleus which is going to divide first and followed by the cytoplasm. So that entire division, which includes the nuclear division along with the cytoplasmic division, is called as the M phase. So this entire division, this is the portion, this is the area in which the cell is going to start dividing. That means in this particular part of the cell cycle, so this entire thing is representing the cell cycle. So what do we understand by the cell cycle? The cell cycle is nothing but a series of events. It is nothing but, let me tell you, it is a series of events which occur inside the cell during cell division. And inside this series of events, there are two major things which are taking place. One is nothing but the interphase, during which the cell is preparing for division, followed by the M phase, which stands for mitosis, and M phase is the phase in which the nucleus is going to divide, followed by the cytoplasmic division. Once the M phase starts, dear students, so here nucleus is going to divide, which is going to get further divided into 4 subphases, in which the first phase of the nuclear division is called as the prophase, followed by the metaphase. The third phase is known as the anaphase, and the fourth phase of the nuclear division is called as telophase. Once the karyokinesis is complete, once the nuclear division has been completed, it will be followed by the cytoplasmic division, which is going to be called as the cytokinesis. So here guys, this entire is the M phase, which is nothing but the mitosis, and before that there is a interphase. Now dear students, you have to understand, since as we said that the majority of the portion of the cell cycle is taken by the interphase, so maximum time is going to be taken by the interphase. Always remember that. So in the interphase guys, remember there are three subphases. Starting with the G1 phase. I am going to tell you each of these phases, and we will study that what happens in each of these phases also. So the first phase of interphase is G1 phase, followed by the S phase, followed by the G2 phase. Correct? So now what we will do is, one by one we will understand what is G1 phase, what happens in the G1 phase, what happens in the S phase, and what happens in the G2 phase. So now the first phase, dear students, as we said that the interphase is divided into 3 subphases. So the first phase is known as G1 phase. Dear students, here the word G stands for gap. So it is also called as gap 1 phase. Since it is before the S phase, and the S phase actually stands for synthesis, it is also known as pre-synthesis phase. So we are going to understand what happens in the S phase also, but before that let's talk about G1. Now what happens in the G1 phase? Now dear students, as we said that in the interphase the cell prepares to divide. So this is the phase which is coming before S phase, and especially in the S phase the DNA is going to replicate. Rather, the DNA is going to double in its content. So what happens mainly in the G1 phase is, whatever the important proteins are required, or whatever different enzymes are required, or the energy is required in the form of ATP, all that is going to be formed in the G1 phase, so that all that can be used for the replication of DNA, which is going to take place in the next phase, that is nothing but the S phase. So that's why, dear students, in this particular phase, as we said, the different types of enzymes, proteins, energy which is in the form of ATP is going to be formed. There will be the synthesis of RNA. As we all know that RNA is formed from DNA, and RNA is important for the formation of proteins. So that's why RNA is also going to get formed in the G1 phase. Now S phase basically, dear students, stands for synthesis phase. Now what is going to happen in this phase is the DNA. Now dear students, DNA is basically nothing but deoxyribonucleic acid, but it is in the form of chromatin fibers inside the nucleus. Now since we all know now that since the interphase is taking place, and now currently if you observe a cell under the microscope in the interphase stage, dear students, you are going to observe inside the nucleus there are certain fibers which are called as chromatin fibers, and these chromatin fibers are nothing but the DNA of the cell. So these chromatin fibers basically are going to double in the S phase. So how is it going to double? That is majorly via the process of DNA replication. Dear students, apart from DNA, which is going to get double, so that's why when we talk about the content of the DNA, as we said that the DNA originally is 2C in content. That means the original content of the cell or the original DNA content of the cell, let's say suppose if it is 2C, then during the interphase, specifically during the S phase, it is going to become 4C. And that's why we said that interphase is important, because once the cell is going to divide, dear students, this DNA that is now doubled up, that has become 4C, it is going to get equally distributed among the daughter cells, and again it is going to get distributed into 2C and 2C. That's why, dear students, we say that mitosis is basically an equational division. So here we are referring to mitosis, and that's why during the mitosis, prior to mitosis, in the interphase, always the DNA is going to get double. From 2C it's going to become 4C. Apart from this, dear students, there is going to be a synthesis of some important proteins in the S phase, which are nothing but the histone proteins. Now, dear students, always remember that histones are very important, because they help in packaging of the DNA, they help in condensing the DNA, because DNA, as we know, that it is in the form of threads, and since the nucleus is extremely small, it is in the size of micrometers. So to pack such a long DNA threads inside the nucleus, the DNA threads have to be condensed and highly coiled. So these histone proteins actually help the DNA threads to become coiled, and that's why the chromatin fibers are able to get packed inside the nucleus, and that essentially is a process which is called as packaging of DNA. Let's talk about the third phase, which is called as the G2 phase. G2 phase stands for gap 2 phase, or we can also call it as the post synthetic phase, because since it is coming after the synthesis phase, that's why we can also call it as the post synthesis phase. Now, dear students, in this particular phase, what happens is, majorly in the gap 2 phase, there is formation of some important proteins like the tubulin proteins. Now, what are these tubulin proteins, dear students? The tubulin proteins are the ones which are important for the synthesis of microtubules, and microtubules are part of the cytoskeletal elements of the cell. So there are three basic types of cytoskeletal elements, which are nothing but microtubules, microfilaments, and intermediate filaments, which actually help in forming the skeleton of the cell. They help in giving mechanical support to the cell. So here the tubulin proteins are mainly synthesized, which are going to be useful for the formation of microtubules. Apart from this, dear students, again here there is formation of certain proteins, as well as there will be duplication of organelles. That means all the organelles which are present in the cell, whether it is endoplasmic reticulum or Golgi apparatus or mitochondria, all these are going to get double in the G2 phase. So in a way that basically, dear students, by the end of G2 phase, the cell has already double all the contents which are present in the cell. And that's the reason why, dear students, we said that interphase is important for the cell, so that all the content, once it is now doubled in amount, now the cell can successfully divide, and it can equally distribute all the content among the daughter cells. So let me tell you one very unique phase which is also part of the interphase. This particular phase is called as G0 phase. Now, what is G0 phase, dear students? Now let me tell you that sometimes in case of certain cells, what happens is, whenever the cell is present in the G1 phase, from G1 phase it directly enters into a phase which is called as G0 phase. Now, dear students, this phase, G0 phase, is a phase in which once the cell enters, it cannot divide further. It cannot divide. That means such cells for the lifetime of the person will not be able to divide, because they will be permanently present in the G0 phase. Such a phase called as inactive phase. And in this particular phase, dear students, the cells are metabolically active. That means the cell is able to form its proteins, cell is able to carry out its own function in the form of different types of reactions which are occurring inside the cell, but the cell is not going to divide. The reason is because the cell is now permanently in G0 phase, it is not going to go in S phase, dear students, and that's why the interphase will never be complete and the M phase will never be started in this case. So cells will permanently remain in this particular G0 phase. Let me give you some examples of such cells. The examples are the heart cells, which are always in the G0 phase. So they also do not divide. At the same time, there are nerve or neurons which are also present in the G0 phase. So dear students, you can also find out if there are any other cells in our body which are present in the G0 cells. If you know any of these cells, you can put the answers in the comment section. So basically these cells are never going to divide in the lifetime of that particular organism, and that's the reason why, dear students, if there is any major injury to this particular cells, then there are high chances that the injuries can lead to the death of that person, because these cells are extremely important and these cells are not able to repair themselves. So dear students, in this today's topic, we have understood the concept of interphase. We understood that what are the three subphases of interphase, that is G1, S as well as G2 phase. After the completion of interphase, the mitosis can start, that is the karyokinesis will begin, which will be followed by the cytokinesis. So I hope, dear students, you have understood the concept of interphase and also the three subphases which are part of the interphase. So see you all in the next video, guys, with some another biology related topic. This is Praful signing off. Take care. Thank you so much. Bye bye.